Hi, I'm Kalaivani Vetrivel, Assistant Professor from SNS College of Pharmacy. So today we are going to see the cultivation of medicinal plants in a country. Cultivation of crude drugs, that is, cultivation of medicinal plants requires intensive care and management. The condition and duration of cultivation required vary depending on the quality of medicinal plants required. We are following many methods for the propagation of the medicinal plant, that is vegetative propagation, that is asexual propagation, and sexual propagation. So coming to the vegetative propagation can be defined as regeneration or formation of a new individual from any vegetative part of the plant body. The method of vegetative propagation involves separation of a part of a plant body which develops into a new plant, and it is having three stages. It is cutting, layering, and grafting. Cutting, these are the parts of the plant stem, root, or leaf which, if grown under suitable conditions, develop new plants. Stem cutting are generally used to obtain new plants. Example: sugarcane and rose, etc. Layering, roots are induced on the stem while it is still attached to the parent plant. This part of stem is later detached from the parent plant and grown into a new plant. Example: jasmine plant. Grafting, new variety is produced by joining parts of two plants. The rooted shoot of one plant, called stalk, is joined with a piece of shoot of another plant known as scion. Example: rose, citrus, and rubber will be involved in the grafting method. And coming to the advantages of the asexual propagations, as resultant species formed through asexual process are genetically identical, useful traits can be preserved among them. And disadvantages is also there. Crops produced through this process have shorter lifespan than those grown through sexual process, and species involved in this process are less likely to resist pest and disease. Coming to the sexual propagations, uh, the process of sexual propagation is pollination, that is pre-germination seed treatments to improve germination rate. This is the transfer of pollen grains from anther to the stigma. Fertilization, fusion of male and female gametes takes place, resulting in the formation of zygote. And seedling, multiplication of plants by using seed is called as seed propagation. Dormancy, it is termed used to describe a seed that will not germinate because of any condition associated either with the seed itself or with existing environmental factors such as temperature and moisture. And rest period is nothing. Some seeds will not germinate immediately after harvest even if conditions are favorable. This failure to germinate is due to uh, physiological conditions. This is said to be seeds or in the rest period. Seed viability and longevity. Pre-germination seed treatments to improve germination rate or chemical acid saccharification and mechanical scarification, and seedling, that is boiled water treatment, soaking in water, moist chilling. Chemical acids scarification is nothing but the purpose is to modify hard or important or impermeable seed covering. Generally, soaking seed in concentrated sulfuric acid is an effective method. Mechanical, seeds of a few species with impermeable seed coat, hard seed coat, can be rendered permeable to water and gases. Their germination is greatly improved. Seedling, pouring boiling water over seeds and getting it to cool gradually for about 12 hours to soften hard shell seeds. Example: coffee. Soaking in water. The purpose of soaking seeds in water is to modify hard seed coats, to remove inhibitors, to soften seed, and to reduce the time of germination. Moist chilling. Seed of many woody trees or shrubs are exposed to low temperature to bring about prompt and uniform germination. Uh, Rate measured by rolled towel test exercise. So coming to the advantages of sexual propagation: simplest, easiest, and the most economical process. Um, Example: some plants, that is trees, vegetables, fruits, can be propagated only through sexual propagation. Example: marigold, papaya, tomato. This type of propagation leads to better crop species that are stronger, disease resistant, and have longer lifespan. Viral transmission and can be prevented in this type of propagation. It is the only propagation process in which resultant offspring have genetic variation and exhibit diversity of characters from parent crops. This genetic variation is responsible for continuous evolution that keeps on producing better and better offspring, easy storage and transportation of seeds. And disadvantages Seeds take a long time to turn into mature plants. That is, time interval between sowing and flowering is longer. Seedlings propagated through sexual propagation are unlikely to have same genetic characteristics as that of parent plants. Some species do not produce viable seeds through sexual propagation and hence are unsuitable to propagate for the same. Micropropagation, plant tissue culture. This method consists of growing cell tissue and organ in culture. Um, Small species of plant organs or tissues growth in a container with suitable nutrient medium under sterilized conditions. This tissue grows into a mass of undifferentiated cells called callus, which later differentiates into plantlets, and these are then transferred into pots or nursery beds and allowed to grow into full plant. Plant tissue culture is widely used to produce clones of a plant in a method known as micropropagation to conserve rare or endangered species. Medicinal plants material should be collected during the appropriate season